Next topic is biological nitrogen fixation. The plants needs nitrogen for their proper growth and maintenance. And we know that in our atmosphere, 78% of nitrogen is present. So plant or some bacteria basically obtain this nitrogen from the atmosphere. So conversion of the atmospheric nitrogen into ammonia, that is NH3, by some bacteria and plant is called as nitrogen fixation. To fix the nitrogen into ammonia, it is very important or it is very necessary to break this triple bond, and this triple bond is highly stable. That's why 78% nitrogen present in the earth atmosphere, because it is highly stable. So to break this triple bond and to make the NH3, we need high energy molecule, that is ATP molecules, that is adenosine triphosphate molecules. So for this is the equation of the nitrogen or biological nitrogen fixation. N2 reacts with 8H positive plus 8 electron and absorbs or use 16 ATP molecules and to convert into 2 molecule of ammonia, 1 molecule of H2, and release 16 ADP adenosine diphosphate molecule and 16 inorganic phosphate, that is Pi. So in this case, the H positive present in the body and electron is also formed, but this electron cannot move freely in the body. So the substance which helps, or the compounds or the proteins which helps in the transfer of this electron in the body of a living organism is called as ferredoxine. Next one is the conditions for the nitrogen fixation. The first condition is that enzyme nitrogenase. Without enzyme nitrogenase, it is very difficult to convert N2 into NH3. The second requirement is the ferredoxine. As we have discussed, the electron is required for 8H positive to convert them into H. So this electron cannot transfer in the body freely. So we need an electron carrier, that is ferredoxine, and ATP molecules to break the nitrogen-nitrogen triple bond, and anaerobic conditions. That means the oxygen should be absent during the nitrogen fixation. The main focus is here is on the enzyme nitrogenase. The enzyme nitrogenase is a metalloprotein, that is, it is made up of the metals and protein. So in enzyme nitrogenase, there are two types of the protein, that is FES protein and MOFES protein. FES protein is smaller protein and it is also called as nitrogen reductase. MOFE protein is also called as molybdenum-ferredoxine protein. Molecular mass of FES protein is from 50,000 to 70,000. FES protein is also called as azoferredoxine, azoferredoxine, and it is a yellow colored protein and it is highly sensitive to oxygen. It do not work in the presence of oxygen. In FES protein, 2 cube of FeS4 units join together. Sorry, FES protein is a cube of FeS4 unit. So supposed to be this is a cube in which there are 8 corners. So Fe and S are present at the alternate position of the cube. So this is our Fe4S4 unit, or I can say that is our FeS protein. The second metalloprotein of enzyme nitrogenase is MOFES protein. MOFES protein is a larger protein of molecular mass 2,20,000 to 2,14,000. MOFES protein is also divided into two parts. The first is called as P cluster and the second is called as MOFE cofactor. P cluster having 8 Fe, 7 S center, and P cluster is having two form. One is the oxidized form and another is the reduced form. Reduced form is called as PN cluster and oxidized form is also called as POX cluster. In POX cluster, 2 FeS4 cubanes are joined together by the sulfur or sulfide linkage of the cysteine amino acid. Supposed to be this is our first unit of FeS4. Here is our SS. This is Fe and Fe. This is first Fe4S4 unit, and there is second FeS4 unit. These two Fe are joined together by the sulfur atom of amino acid cysteine, and in the similar way, these two Fe are also joined together with the sulfur atom of amino acid cysteine. These two cubane of FeSO4 also joined together by a weak bond of the sulfur. In PN cluster, reduced form of P cluster, 2 Fe4S3 units are joined together. So in this case, here it is Fe. This one is also Fe. Here is Fe. Here is sulfur, sulfur, sulfur. then after it is Fe. So this is the first unit of Fe4S3. In the similar way, Fe, Fe and Fe, here sulfur, 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 and finally there is Fe, like this. This is the another Fe4S3 unit. These two units are joined together by sulfur atom of amino acid cysteine, and here also these two Fe are joined together by the sulfur atom of amino acid cysteine. In PN cluster, the fourth sulfur atom is also present in the center of the cavity of this PN cluster in the octahedral geometry. And the last one is MOFE cofactor. In MOFE cofactor, the first unit is Fe4S3 unit. In the similar way, like as Fe, 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 here sulfur, 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 and then it is Fe, like this. And the another unit is MOFES3 unit, that is here is Fe, Fe and Fe. After that sulfur, 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 and in this case there is MO metal, that is molybdenum. So bonding is similar way. This is the MOF3S3 unit. So in this, two Fe atoms are joined together by the sulfur of amino acid cysteine, and here these two Fe atoms are joined together by the sulfur atom of amino acid cysteine. These two Fe are joined together by a weak bond. In the similar way, these two Fe are joined, and here is also joined together by a weak Fe-Fe bond. In the cavity of this MOFE cofactor, there is a less order sulfur atom, which is represented by the Y. Then how N2 is reduced into NH3, or I can say how the enzyme nitrogenase change the N2 into NH3. The main focus of the enzyme is to break the N triple bond N, because it is highly stable bond. This is the cavity of MOFE cofactor. So first the N is trapped in the cavity of this MOFE cofactor, and we know that there is a lone pair present on the nitrogen atom. So nitrogen atom donates its lone pair of electron to this Fe, and in the similar way this nitrogen donates its lone pair of electron to another Fe, because Fe having the empty d orbitals. After the donation of the electron, this nitrogen will become electron deficient. In the similar way, this nitrogen also become the electron deficient. So it pulls the electron from these triple bonds. So this triple bond become electron deficient, or we know that the electron deficient bond is weak. So it helps in the dissociation or in the weakness of this nitrogen triple bond.